Assalamu alaikum everyone. My name is Ayesha Jahangir and I have completed my MPhil in microbiology from the Islamia University of Bahawalpur. It is an honor to present my research at the third International Brain and Biomedicine Conference 2026, organized by Department of Physiology, Government College University, Faisalabad. The title of my presentation is Molecular Detection of Coronaviridae in Rodents from Bahawalpur. The study focuses on identifying the presence of coronaviruses in rodents population and understanding their potential role in zoonotic transmission. Now, the question is how rodents can be the reservoir of coronaviridae. We know rodents are widely distributed mammals and are known to act as reservoir for a variety of zoonotic pathogens, including bacteria, parasites, and viruses. Among these viruses belonging to the coronaviridae family are of particular concern due to their ability to infect multiple host species and they can cause a significant outbreak. Rodents can transmit pathogens to humans through several routes. For example, the contamination of food by rodent droppings, and it is the common pathway. Additionally, the direct contact through bites and scratches can also facilitate transmission. In densely populated areas, especially where sanitation is compromised, the risk of such transmission becomes even higher. Therefore, studying rodents as potential reservoir is essential for better understanding of disease ecology and prevent the future outbreaks. Here is the graphical representation of number of deaths due to COVID-19 for in various countries. The graph represents that coronavirus, like the recent pandemic COVID-19, it has greatly affected US and Brazil. Moving towards the next slide, that is methodology. This slide highlights the global impact of COVID-19 by cumulative confirmed deaths across the different countries. And in this slide, I tried to summarize the methodology adopted to conduct this study. The very first step was rodent collection, the collection of rats. Rats were collected from multiple locations within Bahawalpur. These locations were selected to represent different environmental and human interaction conditions. After the collection of rodents, they were carefully dissected under controlled conditions like proper PPEs and everything. The next step involved the RNA extraction, which was carried out using a Zymo kit to isolate viral RNA from the samples. Following this, the third step was RNA extraction. Okay, and the cDNA synthesis. Yes, the fourth step was cDNA synthesis, and this was performed using the Revert 8 kit. This step is essential because PCR amplification requires DNA rather than the RNA. The next and very most important step is PCR, the polymerase chain reaction, commonly known as PCR. The PCR that was performed was RT-PCR, reverse transcriptase PCR, and it was performed to amplify specific viral RNA sequences. Yes. The final and the last step was gel electrophoresis, and it was used to visualize the PCR products and confirm the presence or absence of viral genetic material. This is a multi-step process, and it ensures accurate and reliable detection of coronaviridae in the samples that we collected from different regions of Bahawalpur. Moving towards the results of the study, a total of 20 sample pools were analyzed, collected from like different locations of Bahawalpur. These slides were tested for coronaviridae and to like total 11 out of 20 pools were found positive for coronaviridae. This indicates a positivity rate of 55%, which is huge. And while remaining 45% of samples were found like negative. Here is a need to elaborate the concept of pool sampling. Each pool consisted of three rat samples, Because we look at the distribution across various locations, we can observe variations in the positivity rates. Here are some of the areas from where we have collected our rat samples, such as University Cafeteria, Qasim Town, and many others, one unit chalk as well. So overall, the results suggest that rodents in these areas may act as the potential causes of coronaviruses. This is the picture of gel showing the positive bands for coronaviridae. So in coronaviridae family, we targeted the RDRP gene, which is highly conserved region in coronaviridae family. Moving towards the next slide, that is scope and importance of this conducted study. Firstly, they highlight the potential risk of zoonotic transmission of coronaviruses from rodents to animals. Secondly, the understanding of host-reservoir relationship helps in identifying how disease emerge and spread within the population of humans. Furthermore, this research will also contribute to developing effective surveillance systems to help from the future outbreaks. Also, in the long-term studies, these are essential for preventing future outbreaks like the recent COVID-19 that was a huge pandemic. So, in conclusion, this study demonstrates the presence of coronaviridae in rodent population of Bahawalpur. It indicates a potential public health concern. And further, more research is needed for better understanding of transmission dynamics and the associated risks. Thank you very much for your attention. I would be very happy to have any question. Thank you.